welcome. Today, we step into the realm where free will ends and pure biology begins. Imagine the human brain as an incredibly complex biocomputer. Usually, it's programmed for social interaction, empathy, and cooperation. But what happens when a bug appears in the source code, our genes, and the environment acts as a hacker that activates this flaw? For decades, science debated, are people born antisocial, or does a cruel world create them? Today, thanks to the work of Avshalom Caspi, Michael Meaney, and Ken Keel, we have the answer. And it's chilling. Psychopathy isn't just a bad personality, it's a tragic resonance between specific genetics and biochemical stress during critical periods of development. We will dissect the process scientists call epigenetic switching and understand why, for some, a conscience is a physiologically unavailable option. Our investigation begins with the X chromosome. This is where the MAOA gene is located, encoding the enzyme monoamine oxidase A. In neurobiology, this enzyme is known as the brain scavenger. Its job is to clear the synaptic gaps of excess neurotransmitters, dopamine, norepinephrine, and serotonin. There are two variations, high activity and low activity MAOAL. In carriers of the MAOAL version, the enzyme works slowly. As a result, the brain is constantly bathed in excess neurotransmitters. You might think more serotonin is good. It isn't. During prenatal development and early childhood, this excess leads to receptor desensitization. The brain gets used to high levels of noise and becomes insensitive to subtle social signals. Hans Brunner's 1993 study first linked this gene to aggressive behavior, but a paradox emerged. 1 in 3 men carries this mutation, yet most are not criminals. Why does the gun stay on safety for some and fire for others? Now we reach the most critical discovery of the 21st century. Genes are not a destiny, they are potential. For a gene to speak, it must be demethylated. Methylation is a process where a methyl group, CH3, attaches to a segment of DNA. Think of it as a molecular lock. If a gene is blocked by a methyl tag, the cell cannot read its instructions. This is a normal regulatory process. But stress turns it into a mechanism of destruction. In 2002, Avshalom Caspi published a legendary study in Science. He proved that carriers of the warrior gene, MAOA-L, become socially dangerous only if they were subjected to chronic stress or maltreatment before the age of 9. When a child lives under constant threat, their HPA axis, hypothalamic pituitary adrenal axis, works at its limit, flooding the brain with cortisol. Cortisol is a red alert signal. If this signal is too high for too long, it triggers a cascade of abnormal methylation. Specifically, it blocks the NR3C1 gene, which creates receptors that are supposed to catch cortisol and signal the body to calm down. When these genes are locked, the brain loses its ability to switch off the stress response. The child stays in fight or flight mode forever. Their amygdala, the fear center, becomes hyperreactive, while the ventromedial prefrontal cortex, the moral control center, fails to develop. Genetic vulnerability and environmental triggers merge into a single pathology. What do we see on an MRI of a person who has undergone this hacking? It's not just a mean brain, it's a brain with dysfunctional connectivity. The key evidence is the uncinate fasciculus. This is a white matter tract connecting the amygdala, emotions, to the orbitofrontal cortex, logic and rules. In psychopaths, this cable is thin or damaged. Research by Gregory et al. In 2012 confirms. When such an individual sees someone else's pain, their mirror neurons might register the signal, cognitive empathy. I understand you are in pain. But the signal never reaches the centers of compassion. The limbic system remains silent. Furthermore, due to anomalies in the reward system, nucleus accumbens, their brain requires extreme stimuli to release dopamine. Ordinary joys, connection, affection, creativity don't provide the chemical reward that a neurotypical person receives. They need risk and dominance just to feel alive. In the brain of an epigenetic victim, the oxytocin system is often compromised. Research shows polymorphisms in the oxytocin receptor gene, OXTR. Where you feel warmth and trust, a psychopath feels only neutral noise. This explains their ability to betray without hesitation. Emotional bonds have no biochemical reinforcement for them. Added to this is a serotonin deficit. Analysis of 5-HIAA levels in the cerebrospinal fluid of aggressive psychopaths consistently shows low numbers. Here, serotonin acts as a stabilizer. Without it, impulses from the amygdala race to the cortex like a wildfire, meeting no resistance. In 2009, an Italian court reduced a murderer's sentence because he had the MAOA gene and MRI abnormalities. This created a precedent. If a crime is the result of gene methylation and amygdala dysfunction, is it a mitigating circumstance? Science tells us a psychopath cannot choose to feel empathy any more than a visually impaired person can choose to see clearly without lenses. This challenges our entire justice system based on free will. Can we fix this? Standard prison or therapy often fails. Group therapy can even make them more dangerous by teaching them to better mimic human emotions. However, hope lies in neuroplasticity. Neurofeedback, training patients to control their brain activity in real time via fMRI, pharmacological unlocking. Scientists are looking for ways to reverse methyl tags, early intervention, the only 100% proven method, creating a super stable environment for high-risk children to prevent end pathological methylation. We are used to seeing the world in black and white, but neurobiology shows us a world of methyl groups and chemical reactions. Understanding these mechanisms isn't about excusing violence. It's about finding a way to stop it before it starts. In the next episode, we'll analyze invisible predators, female psychopaths. Subscribe to understand how estrogen and social masks make their brains even more perfect tools for manipulation.